In this video, we will discuss about bispecific T cell engagers function or how they works. So in the previous video, we have understood what is this BiTEs or bispecific T cell engagers. That is, it is just a combination of heavy chain and light chain of two different monoclonal antibodies, and they are linked together through the linker, which is made up of serine and glycine. And out of these two monoclonal antibodies, one of them is usually targeted around T cells, and generally people use this CD3 protein on the T cells that will be targeted by this anti-CD3 monoclonal antibodies, and the other antibody is targeted to the tumors, which is commonly known as tumor associated antigen. So this could be different and it could be customized based on the individual patient, depending on what are the mutations present in the patients. And in order for this BiTE to work, the essential condition is that this binding of these T cells and the cancer cells should occur simultaneously. Then only this BiTE will work. So once the T cells and the cancer cells interact or attach to this BiTE simultaneously, then it will create a connections that will trigger the T cells activations. And the difference in this case is that it does not require the T cell receptor or co-stimulations to activate the T cells. Just by interactions of this just by interactions of the BiTE to both T cells and cancer cells simultaneously will trigger activations of the T cells, and these activations of the T cells will result in the proliferations of this activated T cells, because once the T cells are activated through these tumor associated antigens, then it's the natural phenomena to activate more to make more activated T cells. So these more activated T cells will then target the cancer cells and then trigger apoptosis. And once the apoptosis is released, then this lytic debris of these cancer cells will trigger other immune cells. So it is actually a cascade effect that will work to enhance the immune response against the tumor cells. But the problem arises in the solid tumors. The problem arises that it's difficult to see T cells and the cancer cells close to each other in the solid tumors, because solid tumors generally have at the higher stage this tumor microenvironment, and this tumor microenvironment prevent this activated T cells, or it will suppress the activated T cells in the tumor microenvironment because of the change in the environmental conditions in the T cells by the cancerous cells. So, however, in the case of like blood cancer such as myeloma, this system can work efficiently. So, if someone can combine this BiTE along with other therapies such as immune checkpoint inhibitor therapies that will prevent the inhibitions of these activated T cells in the tumor microenvironment. So in that case, this BiTE combination with this ICB, that is immune checkpoint blockade therapies, could be one of the viable option to enhance the immune response of the patients against the cancer cells. And so this ICB is not the only option. There are other therapies also that are available that can enhance this BiTE, or other therapies can also enhance the immune response by if they are combined with these BiTEs. So I hope you understood a simple mechanisms how this bispecific T cell engager works and the conditions that is required for these BiTEs to function efficiently. So in this case, the main side limitations of this BiTE is that the challenge is to find the tumors that has this tumor associated antigen, or maximum number of tumors that have this tumor, or let me rephrase it, challenge is to find a tumor associated antigen that is present in maximum number of tumor cells in the patient. If this tumor associated antigen is present in the 70% of the cases, then this BiTE will theoretically work only on the 70% of the tumors. So this is one of the theoretical challenge that one has to face. So if you need to understand more about their functions in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.